Hi, this is Professor Gordon. Today we're going to talk about quantitative traits. So far we've been looking at discrete traits, sometimes referred to as binary traits, that show just two distinct phenotypes. For example, when we were talking about the flies in the virtual fly crosses, we talked about red or white eyes. And if we're talking about disease phenotypes, we might be talking about affected and unaffected individuals, things of this nature. Now, it turns out that many traits show a continuous variation, or at least they show more than two traits, two distinct phenotypes. And examples where that might be are, if you're talking about a flower, the color is white, or it's light pink, or it's pink or dark pink, etc. So this variation may be due to the presence of what we call additive genes. And an additive gene is one in which there's an increaser allele at each gene, which adds to the value of the phenotype by some fixed number. And another possibility is that you see variation due to environmental influences. We're not going to talk so much about that, but it is good to know that such a variation does exist. Also, we're going to talk about quantitative genetics researchers and the fact that they study this continuous variation by using statistical analyses. And it's that last part that I really want to focus on, the statistical analyses. That's what we'll be discussing in what follows. So here's some examples of the quantitative traits. If you consider this moth wing, this particular moth on its wing, here in the upper left corner, you'll see that there are spots, and the number of spots on the moth wing and the size of them is determined by a quantitative trait locus. If you consider the proportion of fur that is dark, in this example that we considered before with the dogs, that's also a quantitative trait. And also down here in the bottom, on the lower left, we have the beak length for different birds. This picture comes from work that Darwin did for finches in the Galapagos. And then finally, this example here in the lower right is just a way of scoring children on their language skills. And you see that these various numbers indicate that children can be scored in anywhere from values from 0 to 4. So that's another example of a quantitative trait because it's not just discrete. Now, when we want to do the statistical analysis, it's helpful to know certain concepts. The first concept is the concept of a histogram, and specifically, how do we construct a histogram? Another important concept is the concept of the mean of sample data. The third is variance, or you can think of as deviation from the mean for a given set of data. And the last is the standard deviation, and that is just actually the square root of the variance. So 3 and 4 are just related by a simple function. And here's an example where we're thinking of height as our quantitative trait for some species. We're measuring height in terms of meters. And we see that the table that we have here on the left, we have 3 columns, x, n of x, and f of x. Now, x is just the quantitative trait value itself. So in other words, we might say, well, if we're talking about I don't know, cats, for example, then we could say that the length of a cat could be, there are cats who are, say, 1 meter long. Obviously, that's a pretty big cat. Or there are cats that could be 1.5 meters long. That would be the second row, etc. Now, one of the things that we want to know when we're doing this is, well, how many cats are of length 1 meter? How many cats are of length 1.5 meters or 2 meters, etc. And you can see in this second column, that's what we do. We just list the number of times we observe an individual with the phenotype value, the quantitative value given in the first cell here, in that row. And then the last thing that we're interested in here is what's called the total frequency for the quantitative value. And for any given row, this value is just the product of the first cell and the second cell in that particular row. So that's just the f of x equals x times n of x. So if we take the example here, the second row, 3 equals 1.5 times 2. And now if we go down to go down to f of x for 10, that's equal to 2.5 times 4, and so on. Now, how do we translate what we have here in this table into this graph? Well, we use bar graphs here in order to do that. And when you're doing a histogram, really the two pieces of information you need are just the first two columns. The third column is useful for computing variance, but for the time being, we're not going to use it for creating anything having to do with the histogram. So let's take a look here. So on my horizontal axis, I'm going to list all these possible x values. You'll see that I have 1, 1.5, 2, 2.5, 3, etc. up to 4.5, and that's what you have here, 1 in the table all the way down to 4.5. Now, the height of each of these horizontal markings or quantitative traits is just the frequency. So, for example, you see when you have 1, that n of 1 is just 1. So you see here in this bar graph, it just goes up to a frequency, a vertical height of 1. If we go to say 2, which that has three individuals have a quantitative trait height value of 2. So if we go to 2.0 here on the right, we go up. You see that goes up to 3. And similarly, let's take 3.5. You'll see 3.5 has 3 values there. So if we go over here to 3.5 on the horizontal axis, you'll see that that goes up to 3. And likewise for the others. Now, we said that we wanted to calculate the mean. That's the second thing we want to be able to do. And to calculate the mean, we can use the table that we had previously, but now we're going to use all 3 rows, rather all 3 columns. And so to calculate that, we take the sum of the total frequencies, which is the sum, this summation, this Greek sigma means sum over all the possible values of x, of f of x, and that just means take the sum of all the cells here in this column. Okay? And then you divide it by the sum of all the individuals, which is just the sum of this column. So if we sum up all these values here in this column, you'll see we get 55. And then for the n, you'll see if we sum those all up, we get 20, meaning there are 20 individuals that have been sampled. So we're looking at 55 divided by 20, and that's equal to 2.75. So the mean height in this group of individuals is 2.75 meters. Question 1. If we add more individuals with heights of 4.5 meters to our sample, what will happen to the mean? I.e., will it increase, decrease, or stay the same? Provide an example calculation starting with the data from the previous slide. Okay, so we've calculated the mean as 2.75, and you can see here that because of the data that we chose, it was symmetric. Not surprisingly, the mean is right in the middle of the distribution of quantitative heights here, which is, shall we say, unusual, but it's a good starting point when we're teaching the subject. Now, if we calculate the variance, which is, as we mentioned, the spread, to do that we have to first find the difference between each of the values, the quantitative trait values, and the mean. So remember that the mean is equal to 2.75. So now I've added 3 additional columns here in the table, and I've removed the frequency values in this table. And so the way we do that is we just look literally at the difference of the mean minus of the quantitative trait value, and we call that D. Okay, and then we square it. And then the last column, we multiply the sample size, which is the total sample here, times d squared. No, I'm sorry. It's this times d squared. So it's n of x. Okay, so let's do the first one. The quantitative trait value is 1 here, right? In this cell, as indicated by this cell, there's one individual who has this value. The difference between the mean, which is 2.75, so 2.75 minus 1 here, that equals 1.75, right? Well, if you square 1.75, you get 3.06. And then when you multiply 1 times 3.06, you get 3.06. Now let's consider the second example, 1.5 quantitative trait. We have two individuals who have a quantitative trait value of 1.5. In other words, they're 1.5 meters in height. And if we look at 2.75 minus 1.5, we get 1.25. Then we square that, and we get 1.56. And we multiply 2 times 1.56, and we get 3.12. And so on and so forth for the remaining rows in our table. Then when we sum up these terms, n of x times d squared, we get 16.2. And we have a total sample size, as we mentioned previously, of 20. Question 2. Sum up the column in the previous slide labeled D. What value do you get? Use this information to suggest why we use the value D squared in the calculation of the variance and not D. All right, to calculate the variance, what we need to do is we need to take the sum of the last column from the previous slide and divide it by n minus 1. So in this instance, it'll be 16.2 divided by 20 minus 1 is 19, and we get 0.85. Just as an aside, that n minus 1 refers to, you may wonder why we don't divide by n, and it turns out that it has something to do in statistics with something called bias. We want an unbiased estimator, and so we have, in order to achieve that, we have to divide by n minus 1 instead of n. But that's not something that you need to worry about in this course. So as we getting back to the calculation of 0.85, it means the variance or spread around the mean is 0.85 meters. But we don't think in terms of height being in squared meters or m squared. So how do we fix that? Well, I will mention that in a second, but first I want to just say, remember that the variance is the spread around the mean, but distributions with the same mean can have very different variances, and that explains why it's critically important not simply to just calculate the mean when you're doing these calculations, but also to compute the variance about the mean, because as you see, you can get very different shapes, meaning you get different distributions, and in a sense, you're getting different histograms. So your histograms are changing, and as a result, that can alter what your accounts are going to be in various quantitative trait categories. So because we don't think in terms of n meters squared, we need to take the square root of the variance to work in units that make sense, or meters. So we take the square root of 0.85, and we get 0.92, and that's our standard deviation. So one standard deviation from the mean is 0.92 meters. And you can see in this figure here on the right that for a normal distribution anyway, or a bell curve, that about 70% of your observations, if you were to sample from such a distribution, about 70% of the time they would be within one standard deviation. About 96% of the time they'd be within two standard deviations, and 99.7% of the time they'd be within 3. So let's talk about some genetics here. You recall we were talking previously about the additive effect of alleles. Each allele adds a fixed value to the phenotype, so it might be color or size or number. That is your quantitative trait phenotype. And so if we consider an example for a hypothetical flower, a color phenotype, suppose that I have two alleles, big R and little r, And big R is the increaser allele. So that gives 1 unit of red pigmentation, while the null allele, or little r allele, has no effect at all. Baseline is homozygous little r, which is just a white flower, or you could say a colorless flower. If I'm heterozygous, I get a pink flower, and if I'm homozygous big R, I get a red flower. You'll notice this differs slightly from the example we did with a dominant and recessive, in the sense that the heterozygote doesn't look like either of the two parentals, but that was not the case when we dealt with a dominant and recessive. So if we're talking about polygenic inheritance, what we mean by that is more than one gene can affect the phenotype, and each dominant gene adds to the phenotypic value. And what they mean by dominant gene here is each increaser allele adds to the phenotypic value. So an example involves human skin pigmentation. That's affected by 3 genes, and each allele is additive. So we see more classes of phenotypes in this way, and you'll see that the shading here in this distribution represents what you would expect to observe with regards to quantitative traits and, or skin pigmentation in individuals drawn from the population. So here's an example of some exercises you're going to see. Suppose we have height in corn plants, which is controlled by 4 loci, A, B, C, and D. Now, individuals with a multilocus genotype of little a, homozygous for all the non-capital letters, so little a, little b, little c, little d, those individuals are all 20 centimorgans long. Those individuals who have the maximum genotype, multilocus genotype for length would be homozygous big A, homozygous big B, homozygous big C, and homozygous big D. Now we can ask this question: how tall is a plant with the following multilocus genotypes? Suppose it's heterozygous at the A allele, locus, it's homozygous for big allele for big B, it's heterozygous for big C for the C locus, and it's homozygous for the increased allele D at the D locus. Well, the way we do this is, first thing we do is we find our baseline genotype, and the baseline is little a, little a. It's little e, little b, little c, little c, little d, little d. And that's 20 centimorgans, right? So that's the smallest that any corn plant could be. Now we compare it with the maximum genotype's height. So the maximum multilocus genotype is one in which all the alleles are capital. So all genotypes are, add 1, or they add a fixed unit to the amount to the height. So that happens to be 60 centimeters, right? So to calculate the height difference between the two, to figure out the height, we have to use this formula. This formula actually is just the famous slope formula when you're talking about lines. So you may recall that it's y1 minus y0 over x1 minus x0. y1 is the phenotype value, so the maximum for the maximum that's 60, and for the minimum that's 20. And then the denominator is, well, x1 minus x0, but x1 just refers to the number of capital alleles in my multilocus genotype. So for here, that's 1, you can count it. It's 8, right? A, B, B, C, C, D, D. And then for this multilocus genotype, it's 0. So I have 8 minus 0 in the denominator. And that gives me a value of 5. I get 40 over 8, which equals 5 centimeters from each increaser allele is the contribution to the overall, it's the contribution. So every copy of a capital letter in your genotype adds 5 centimeters to your to the length of your corn plant. If we're considering the example here of big A, little a, big B, big C, little c, big D, then we can compute that that in fact is 50 centimeters tall, because our baseline, remember, when you have no increaser alleles, is 20, and every copy, this big A, this big B, this big B, this big C, this big D, this big D, contributes 5 centimeters. So that's, you have 6 of them, so that's 5 times 6, which is 30, and 20 plus 30 is 50. Here we have it represented in a graph. So here the vertical axis is the actual QTL value, and you remember it goes between 20 and 60. And then on the horizontal axis, I have the number of increaser alleles in my multilocus genotype. So you can see, for example, here at 0, well, there's only one multilocus genotype that has 0 increaser alleles in it, and that's this one, the little a, all the way down to little d. And sure enough, that has a quantitative trait value of 20, as we talked about in the previous slide. Now, if I'm talking about the situation where I have one increaser allele, well, that would be, there are a number of different multilocus genotypes that can have that. But basically, the story is that there can only be one capital letter in the string of letters, and I've indicated those here by the blue. Now, the one we're interested in, the question was about this particular multilocus genotype, and I've indicated those letters that are capital by green. Okay, so n equals, that means that n equals 6, and if you just draw the line up from n equals 6 here, you'll see you get to 50, right? And that's the corresponding. It turns out, for example, this multilocus genotype also has 6 increaser alleles, and so does this one, and so they also would have a quantitative trait value of 60 centimeters.